From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 56, recorded on November 25th, 2024. I'm Vincent Racaniello, and you're listening to the podcast about the nervous system. Joining me today from Salt Lake City, Jason Shepard.
Morning, Vincent.
How's it out there? You got cold weather yet?
Yes. Well, actually, my house was 11 degrees this morning.
Inside?
No, not inside.
Outside?
That would be good.
Is there snow in the mountains yet?
Yeah.
Cool.
Couple of storms already.
So you can go skiing, right?
Yeah. The most of them are open, I think, this week.
Nice. Also joining us from New York, Tim Chung.
Hello, hi. Jason, when you said 11 degrees, is that in Fahrenheit or
In
Celsius?
Fahrenheit.
Oh, okay. Because I was
Yeah.
like, oh, that's beautiful, because that's what we have in New York, but that's in Celsius.
No, Fahrenheit, which I think is like minus something in centigrade.
Yeah.
We have 11C in New York, yeah.
Yeah, it's beautiful. It's
Wow.
wonderful outside.
It's mostly sunny, yeah. Although it's supposed to rain tomorrow. I think a couple of announcements. First of all, if you like these programs, we'd love to have your support. In fact, this is our fundraising season, November and December. Science is under siege in this country, as you know. A lot of anti-science rhetoric in this new administration. So we're going to be here to give you good science. And we need your support to do that because we don't do ads. We are supported by listeners. So please go to microbe.tv/contribute. We have some nice incentives there to incentivize you to give at certain levels. And if you are concerned about science going forward, in particular, the nomination of RFK Jr. as head of Health and Human Services. Now, this is an agency charged with taking care of America's health, okay? And he has spoken out against vaccines many times. His policies have led to people dying in Samoa of measles. And he claims he wants an 8-year pause on infectious disease research, which is just dumb. It means you don't know anything. How can you do that?
What's the justification? He thinks that
he wants
this
diseases
political
are
witch
going on
hunt.
pause.
He wants to focus on chronic diseases, supposedly. But
what if some of those chronic diseases are triggered by infectious disease?
Yeah,
Yes, exactly right.
Yeah, long COVID is just the tip of the iceberg. Probably
He doesn't get it. He does not get it.
Yeah,
but also, this is against vaccines and Tony Fauci and all that. Anyway, I have a website for you. The key here is to write your senator and say, please do not confirm RFK Jr. for this reason. We made a video here at Microbe TV that summarizes some of his ridiculous stances, anti-science stances, if you want to go. It's on the YouTube channel. But the website is safecommunitiescoalition.org/RFK. And you go there, though. They have already populated the form with a form letter, but you can change it, put in your address, send it to your senators. It's really important to speak out. Your voice matters, okay? Okay, so today, I can't remember, though. I think we're doing a paper on memory.
We are, which, obviously is one of my favorite topics. And I mean, my plug is, we need to understand how things work before we understand how things break. And a lot of the research that we discuss in this podcast is basic fundamental science that we try to explain and try to sort of, talk about their implications for disease and human health. But without any of these basic science papers or work that comes out of government-funded and taxpayer-funded work, there's no breakthroughs in therapy. There's no translational research. So that's my plug. Anyway, so learning and memory, we're going back to a couple of topics that we've covered over the years. One is, how do you store information in neurons in the brain? And we've always thought that information and encoding, storage, recall, all the things that we associate with memory are just neurons. Neurons are the cells that are firing and processing the information. And for a long time, we've just ignored the other cell types in the brain. And in the last 10 years or so, these glia cells have become much more studied. And, there's been a lot of sort of dogma overturning kind of research, partly just because the dogma that was established was ignorant of what exactly these other cell types are doing in the brain. So
they're just glue.
What's that? They're just glue. Yeah, glia meaning glue, meaning they're just there to support the neurons, or and to some degree that is true. They are there to keep neurons alive, and they're a lot more dynamic and a lot more involved in all of these processes than we anticipated or that was thought. So the paper we're going to discuss today is called Learning Associated Astrocyte Ensembles Regulate Memory Recall. And this was published in Nature, 6th of November. First author is Michael Williamson, and this is from Benjamin Deenan, his lab at Baylor College of Health. So, lots to break down here. Learning associated astrocyte ensembles. So astrocytes, what are astrocytes? The astrocytes are one of the major glial type of cells. We've covered them a little bit, I would say. We've talked about microglia as well, but astrocytes have been known to be integral for synapses. So they actually physically contact synapses, and there's this idea of this tripartite synapse, and this is not. This is part of the presynaptic and postsynaptic side of things, and in the end, it seems that the astrocytes are very important for actually regulating the structural part of these synapses. So this is not always, something that we're talked about, and we're not going to have time to go into the details of how the astrocytes. But the major idea coming into this was that they thought the way astrocytes regulate synapses are sort of passive, as in you get presynaptic neurotransmitter release, and the astrocytes help buffer the amount of neurotransmitter in the cleft, so that you get this sort of fine tuning of presynaptic neurotransmitter activation of the receptors. And that was thought to be, sort of there to tune things. But the astrocytes themselves were considered not to be active, as in they're not firing action potentials, and there's no sort of changes in the voltage of their membranes. So they're not actually thought to sort of contribute directly to the processing of the information. They're just sort of there to buffer the synapse. But over the last few years, there's been more and more evidence that there's calcium signaling in astrocytes that seem to be in tune with the firing of neurons. And one of the sort of the things that comes out of this is that if there's calcium activation or calcium signaling, we know that calcium is a super important second messenger that induces second messenger cascades that signals in most cells. And so in neurons, we know as well that not only do you get these electrical, firing of action potentials, but for memory, especially, you also induce gene expression when you learn. And so there's a cascade of genes that get turned on when you learn, and the first wave of gene expression is very quick, and it's thought to be calcium dependent and activated through the receptors at synapses. And so we've talked about some of these genes that are turned on immediately with learning. Like my favorite is ARC, but there's also CFOS. And we talked about FOS as being a transcription factor, so it gets activated in neurons with learning, and then because it's a transcription factor, it turns on other genes. But for the memory aspect, these immediate early genes have been used to tag the cells or the neurons that are active during learning. So that has sort of led to this idea called the engram, which is the circuit, the memory circuit that is important for both encoding memories, but also storing and, of course, then being able to recall the memory. And so the simplistic idea here is that you've got a set of cells, set of neurons. I keep on saying cells because I have to include not just neurons. Neurons that are active during encoding, and then that they are then important for processing and making the memory engram. And there's a whole bunch of sort of plasticity going on to do that.
Is there a specific part of the brain where these neurons reside?
Yeah, so at least for memory encoding, we think it's the hippocampus, so the cells in the hippocampus, and that's where we're going to be looking at here as well. So that's just all to set up the idea that there's gene expression with learning. There's a set of cells, a set of neurons that are active during that learning episode.
And isn't correct me if I'm wrong, but is the idea of the engram is that if you remember, say, context A or thing A, the set of neurons that are lit up or are firing is different from the set of neurons that would be firing if you remember context B or the thing B. So different things have their own different engrams or set of neurons.
Right.
And that's how your brain tells different things apart.
So the neurons don't double up?
Well, so they do. So most of the tagging approaches that have been used, they light up the cell, so the whole cell. But cells, different neurons can be involved in different engrams, and it just depends on a lot of so there's actually a time dependency here. So if you have context A and context B very close together, there is some overlap with the cells, the neurons that are tagged. But the idea is that if you were able to look at the synapses, there's specific synapses on each neuron that would encode the engram. And so it makes sense that since you've got at least a couple of hundred synapses on one neuron, that it'll be involved in different engrams. And it's the specific connected circuit that differentiates the different engrams.
So let me ask you, how many neurons do you need to remember your phone number?
Yeah, well, so some theories think that there's a single neuron that encodes, your grandmother, or these sort of very abstract memories. But yeah, we think it's probably more than a single neuron, that there's a set of cells. When we tag these neurons during encoding, they probably tag about 5 to 10% of all the neurons in the hippocampus. But if you actually go back and then do a recall test and you see what the overlap is, it's only about 5% of the 5%. So in the end, it's a small set of neurons, but it's not just one.
Okay. Actually, things like phone numbers it's short-term memory. You can remember a phone number. It's about the maximum length you can remember in a short term, right? So someone gives you a phone number, you can write it down in a few minutes, right?
Right, and that's called working memory. So, which is actually interesting that humans have this sort of cap of how many objects or numbers they can remember in their working memory, and then the long-term consult. So if I come back and say, hey, did you remember this phone number that you tried to memorize yesterday? You have to actively turn that into a long-term memory. So the working memory, and usually that's also why if you're sort of holding someone's name in your working memory or a phone number and someone interrupts you or you just lose attention, it's gone.
Right.
You have to actively think about storing that or memorizing it. So all of these gene expression pathways, as I mentioned, were thought to be specific to neurons. And so the main hypothesis and sort of initial sort of investigation in this paper is that they were like, well, what about astrocytes? We know that there's this calcium signaling. We know that they seem to be actually more involved in learning and memory than we thought. Do they actually turn on genes when you learn? And if so, can we tag the astrocytes in the same way that we did for these neuron engrams? And so that's what they set out to do. So they used similar approaches and technology that had been used for neurons. And the first thing they wanted to address was, do you see immediate early genes expressed in astrocytes after learning? And they concentrated on CFOS, and indeed they do. And this has not been this is not brand new information. There has sort of been hints that this is the case in astrocytes, but this is the first.
Do you think you see the rest of the family of immediate early gene, or is it just FOS?
Well, we never see ARC in astrocytes, at least transcribed. Okay. So I think it's not all immediate early genes. And so that specificity is interesting in itself. And some of these, for ARC, it doesn't surprise me too much because we think that ARC, to affect a protein, it's not a transcription factor. It affects synapses. Astrocytes don't have synapses, so why would you put ARC in there? But for FOS, it's a general transcription factor, and in fact, FOS is expressed in throughout the body. It was actually initially discovered in fibroblasts, and it's because it gets activated by growth factors. So some of these transcriptional processes are, generalized to other cells. So perhaps this is not too surprising that FOS is one of those immediate early genes that gets induced with learning. And so they're looking at astrocytes in the CA1 region of the hippocampus, which we know is, important for these memory encoding events. So when they observed that, since the endogenous FOS is induced, we thought, well, let's try to tag these astrocytes using the FOS promoter. And instead of expressing their tagging plasmids and neurons, they drove expression in astrocytes by using an astrocyte promoter called GFAP. And they also wanted to know if FOS expression in astrocytes was actually important for anything. And so they sort of did a couple of things here. So the first experiment, they took FOS conditional knockout alleles. So these are alleles that have these Cre-Lox sequences. And so the mouse has this transgenic Cre-Lox sequences, but they're only active when you express this Cre protein, and the Cre protein then allows the recombination to delete the sequences in between those lock sites. So you can get a conditional knockout of your gene in whatever cell type that you want to actually do this in. So they use this Cre expression in under this GFAP promoter. So they're only trying to get rid of FOS in astrocytes. And so they show that they can do this, and when they do this, they get a nice decrease in FOS expression, specifically in astrocytes, but not in neurons. And so then they wanted to look at whether this affects anything that's important for memory. And the first thing they looked at was synaptic plasticity. So again, these are in astrocytes. So they're just looking at knocking out FOS in astrocytes. And now they're going to record from neurons to see if there's any changes in neurons. And we've covered this, I think, a couple of times, but one of the models for how the engram gets formed is that you get this synaptic plasticity. And one of the models of strengthening synapses is called long-term potentiation. And so they took slices from these conditional knockout mice, hippocampal slices, and they record from the CA1, and they can induce LTP using a particular kind of frequency of neuronal activation. In this case, it's called a theta burst stimulation. And what they saw was that in these FOS conditional knockouts, again, just in the astrocytes, they actually don't, they have a huge deficit in LTP. So this is the first surprise here, that you've got this non-cell autonomous effect on neurons from astrocytes that would normally express FOS. You get this LTP deficit. So you can't, you induce LTP, but it seems to decay very quickly. This is in figure 1E. So that's a surprise. They don't actually really come back to this observation in terms of mechanism. So we still don't really know why you would need FOS in astrocytes for LTP, but they kind of speculate a little bit.
They did do one experiment that's quite interesting, which is initially they were able to induce FOS in astrocyte by stimulating neurons in the hippocampus. So when the hippocampus fires, maybe caused by learning, the astrocytes also fire or like induce FOS. And they inhibited the input to the hippocampus using like a retrograde AAV that inhibits input. And they then stimulated the postsynaptic neurons. And yet again, they saw FOS induced in the astrocyte. So it tells you a little bit that it is actually the postsynaptic side that is talking to the astrocyte. Because initially I thought, it's a glutamate release on the presynaptic side. But maybe it's not. So it's kind of, that I found quite surprising, that it is actually the postsynaptic neurons telling the astrocytes, perhaps.
Right. Yeah. I mean, and so that, so that signal, so it's this complex interplay between the astrocyte and the neuron. So I'm not too surprised that you need neurons to activate the astrocytes. But it is interesting that, as you mentioned, that maybe it's through postsynaptic side. But then you're activating the astrocyte, but the astrocyte then is doing something else to regulate synaptic plasticity on the postsynaptic. So there's this interplay between the two. On.
Yeah, but it makes sense, because I remember people telling me, like, if you were to grow neurons on the disk, the neurons would form connections to each other inside the disk. Only if you have astrocytes also growing in the disk. If you remove the astrocytes, then the neuron synaptic formation in the disk gets massively removed.
Yeah, I mean that's true to some degree. I mean you can get synapses forming without astrocytes in a dish. But it was always thought that actually the astrocytes are releasing goodies that are trophic factors that are important for building the synapse. But once you have the synapse built, we just thought the astrocytes were passive and they were buffering the synapse. They weren't thought to contribute directly to these sort of very dynamic changes in synapses and fully formed synapses.
Okay.
So you're absolutely right. Like, we always thought that astrocytes were important for building the synapse during development, but after that, they were just there to support them.
Maybe we're always in Japan.
Could you talk briefly about how they're measuring memory and what kind of assays they're doing in mice?
Yeah, so this plasticity, synaptic plasticity, is in a slice, but the next thing they wanted to do was to actually look directly at memory, and so in this case they're using this contextual fear conditioning assay, which we've talked about it quite a few times, where you put a mouse in a box. The box has some stimulus, visual or olfactory, that makes it specific, and then the animal gets shocked in that context. So that's context A, for example. And this has become the most well used memory assay, just because it's very easy. It's a one-shot learning, so you can put the animal in that box once. It gets shocked once during that learning episode, or it gets shocked more than a couple of times, but only in that one sort of event. And then you come back a day later, and the animal shows that it recalls that it got shocked in that box, even when it's not shocked, because it freezes, which is this automatic innate behavior that the animal has when it's scared. So it's a very quantifiable response. It doesn't need any fancy technology. It's literally just, does the animal move or not? So that's what they did actually in the next experiment here, where they inject those AAVs, the viruses that will express the Cree, just in the astrocytes. They wait for weeks. They train the animal with this shock, and then 3 days later they come back and they put the animal in the same context box to see if it recalls. And so what they found was that the animals freeze normally in the box before they get shocked. So there's no sort of anxiety or changes to how much the animal will freeze when you put them in the box. But after recall, they did see a deficit in the amount of freezing in these FOSS conditional knockouts. Now, the result's significant. It's and I would say this is what you would see in some cases where there's some deficit in memory recall, but it's not complete loss of recall. But it's quite it's substantial. So I think along with that LTP observation, it's very interesting. And again, this is just fast knockout in the astrocytes. They also showed that if you, they also had a final assay there. So there's this contextual fear conditioning, but then there's another simple assay called novel object recognition or novel place recognition, where you have two new objects in their cage that you put in there, and mice, just like humans, are curious, so they'll explore the new objects. But if you come back a day later and you move one of the objects, the mice think, oh, well, what happened? What's happened here? They moved the object. This is a new thing. So they explored that moved object more than the other object that was not moved. And so they show that this discrimination, so remembering that the object was moved, there's a deficit in these FOSS animals as well.
So, by the way, the FOSS knockout, that's done before the conditioning, right?
Yep.
Would it have any effect if you did it afterwards?
Yeah, so that's a good question. They did not do that experiment, so I would like to see that.
They do one maybe in the later figure. They do a little bit, because right now we can't really tell if this FOSS knockout affects memory kind of formation or memory recall.
Right. Yeah. So we'll get into that a little bit more detail later for sure. Okay. So right now we know that FOSS is induced in astrocytes. If you take out or prevent FOSS from being induced in astrocytes, that seems to contribute somehow to memory in mice. So then they were like, okay, well, let's try to see if there's an astrocyte engram, an astrocyte. Well, they're calling it an ensemble. There's a lot of sort of debate of what to call these things. Ensembles, engrams, they're sort of the same thing. So here they used again a system that's kind of neat. They use the FOSS promoter now to make their tag activity dependent, and then they use another construct that will express TD tomato only when the FOSS promoter is activated. And so in this case, they're using something called flip, which is similar to Cree. It also excises things. So now you've got this double sort of intersectional way of turning things on and off. But essentially, what happens is that there will only be TD tomato expressed in the astrocyte if it's induced by activity through the FOSS promoter. And so that's the first thing they show, is that indeed, if you take a mouse and compare the expression of TD tomato in the hippocampus and the astrocytes in the home caged animals versus the animals that underwent that learning paradigm, the fear conditioning, there's an increase in expression of the TD tomato in these astrocytes. So that suggested that indeed you could probably try to manipulate now the astrocytes that were turned on. Because before, with the FOSS knockout, that was all FOSS was knocked out in all the astrocytes. So now they're trying to get at just the activity-induced FOSS expression or astrocytes. So in the next experiment, so now they're saying, okay, we can label these astrocytes. What can we manipulate them in some way? And here they actually use another sort of system to drive GCAMP, which we've talked about, which is this calcium sensor. And they wanted to know, is there something happening in the astrocytes that were induced? Is there some sort of difference in their properties? And so they expressed GCAMP just in the in response again to the FOSS promoter. So they looked at home cage versus fear conditioned, and what they noticed was that there was more calcium activity in the set of cells, the set of astrocytes that were active, that were induced by the FOSS. So that's kind of similar to what you expect to see in neurons, that there's some sort of change in their neuronal activity, and so again suggests that the astrocytes are directly communicating with the neurons, and now there's some sort of change in those astrocytes that were induced or activated during this learning episode.
I wonder if they didn't say anything about calcium waves, and I wonder if the astrocytes that are part of this ensemble are preferentially waving at each other calcium-wise.
They didn't talk about calcium. I mean, the calcium waves in astrocytes has been somewhat controversial, and some people, some have seen it, some have not. So maybe they want to just stay away from that. So how the astrocytes are, it's thought that actually astrocytes are connected directly with each other through gap junctions, not synapses. And so gap junctions are sort of just basically holes in the membrane that allow ions to flow directly from one cell to the other. And that in some cases you can get this sort of calcium wave going through a number of astrocytes because of these gap junctions. So now they've got their astrocytes tagged that are induced by learning, and so now they want to know, can we actually tag both the neuronal engram and the astrocyte engram at the same time to see if there's some sort of relationship between the two. And And so again, they use a bunch of fancy genetics here, where they now express both a neuronal promoter and an astrocyte promoter under the influence of FOS. So the FOS only activates, so you'll get expression in neurons and astrocytes only if FOS is activated. And so they can get nice labeling now of the the neurons that are active during learning. So GFP is expressed in the neurons, and the TD tomato is expressed in the astrocytes. And the first thing that they wanted to know was, is there a physical relationship between the astrocytes and the neurons? So one could expect that maybe the astrocytes that are induced are preferentially closer or physically contacting these engram cells. And that seems to be the case. So they quantified the coverage, the co-localization basically of the astrocytes with the neurons that were labeled, and there is a preferential increase in the TD tomato expressing versus the non-TD tomato astrocytes.
And the TD tomato being the if an astrocyte expresses TD tomato, then it has been is part of the engram.
Right.
It's the engram marker.
Right. So then exactly.
What's interesting is that all the astrocytes that are, well, maybe not all, but a lot of them that they show in the pictures, the astrocytes are induced. They're induced in the hippocampus. All these pyramidal neurons, they're excitatory neurons. They have these like really nice apical dendrites shooting up in one direction, and that's, I'm guessing, where all the input comes in. And all the astrocytes seem to be where all the dendrites of the neurons are. So this is quite consistent with them regulating, physically where the plasticity is taking place.
Right.
You see what I mean? Because originally I thought like maybe they would be around where the cell body is, perhaps. Maybe it's regulating something there, but no, it's actually all at the dendrites, where the plasticity is, expected to take place.
Yeah.
So yeah, it's quite
and that's actually, kind of what so the next experiment, they wanted to try to, so they see this physical coverage of the activated astrocytes with the activated neurons, but what about synapses? And so here they used, again, a sort of complicated genetic approach, something that I think we may have covered. It's a technique called GRASP. So essentially, they're expressing a split GFP, and the split GFP only fluoresces when both sides of the GFP are physically close to each other. And so they expressed one side of the GFP in neurons, and they expressed the other side in astrocytes. I think they expressed the one side of the GRASP GFP in the presynaptic neurons. And the other side in the postsynaptic neurons, postsynaptic neurons. So it forms, it actually is looking at the CA3 to CA1. Right,
connection.
CA1 to CA3. Yeah, thanks.
Whereas the astrocyte is like expressing TD tomato.
Right.
Yeah. So they're asking the question whether the puncture formed by the GFP being connected at the synapse is that close to where the tomato is, where the astrocyte is.
Right. So it's not the astrocyte, which wouldn't make sense. But basically now they're saying this is trying to get at the synapses that are activated during this learning episode. So it's the CA3 to CA1, and I don't quite understand, so this GRASP technique has been very difficult for many to get to work. They sort of don't talk about, they sort of rely on previous publications to say that these are the activated synapses or the engram synapses. And there wasn't a ton of controls here to really show that is the case, in my opinion. But anyway, there has been some papers again that have used this approach to say this. So they then show that indeed the TD tomato astrocytes preferentially seem to overlap with these these GFP positive synapses. So again, suggesting that there's some relationship between the synapses that were active during this learning event and the activated astrocytes.
Have you thought about, I wonder if the authors or if you have thought about whether each astrocyte would regulate the synapses for one engram, or would it be regulating the synapses of multiple engrams? Because by counting how many astrocytes you have there, maybe normalized to how many neurons you have, you can get a rough estimate of how many possible engrams a mouse can have.
Yeah, so I mean, I think there's a lot of questions here that are sort of interesting, right? So astrocytes, if you look at the number of synapses one astrocyte can contact, it's hundreds, lots. In fact, there's also this idea that human astrocytes are larger, they could cover a lot more ground. So how you would get specificity of contact with these synapses is not clear. So there must be some signal. It's that the possibility is that they're dynamic, and that there's certain parts of the astrocyte that are activated in a local region.
So maybe there are more. So if you look at the neuronal engram between two different engrams, so like two different types of memory, maybe the overlap between the two engrams or neurons would be more specific, so less overlap. Whereas for astrocytes, maybe there'll be more overlap, perhaps.
Right.
Okay.
Okay, so the next experiment now is so they show that this, and I should have mentioned like this, the way they induced LTP before was to stimulate CA3 neurons and then record in the CA1 neurons, and this is a very well-defined pathway. And so they actually now, it's kind of clever, they wanted to say, okay, if we go and artificially activate the astrocytes independent of the neurons, can we induce LTP? So is it sufficient to do that? And so they targeted the astrocytes with this GPCR, this G-coupled protein receptor that has been used to influence calcium dynamics. It's called a DREAD. So this is like this designer receptor that's only activated when you have a drug that's exogenous. And so these receptors are silent until you add the drug, and then the drug activates the receptor. So they express again this receptor in the astrocytes, and then they actually did made some hippocampal slices, and they added this drug, and they also sort of looked at whether this was in the sort of so-called engram cells, the T tomato versus the non-engram cells, whether this activation of the astrocytes influenced synaptic strength. And they did that by measuring these many EPSCs. So they're now patching individual neurons, and they can record these minis, which are very sensitive to neurotransmitter release and receptor activation. And what they saw was that when they did this in the engram cells, in the engram astrocytes, you got an increase in the frequency of these minis, which has traditionally been thought to be a presynaptic effect. So now there's more neurotransmitter being released, and it is somewhat correlated with the strength of the synapse. It was interesting that they didn't see a difference in the amplitude of these minis, which is thought to be the postsynaptic side, the receptor side. But anyway, so again, it suggests that that they didn't see this effect in the non-engram astrocytes, so the non-activated astrocytes. So something happens in these astrocytes that are turned on with learning, and they can actually talk back to the neuron directly to influence synaptic strength.
And this is after the memory's been formed. You can still alter downstream kind of communication between neurons, even after the synapse has already been potentiated. Yeah.
Right.
Presumably.
Exactly. So it sort of suggests that there is some sort of physical alteration, physical contact or long-lasting change in these astrocytes that are affecting the neuron. Okay, and so then the final sort of big experiment they did was coming back to how do these astrocytes actually get involved in the memory itself? So is it, as Tim said, formation? Is it storage? Is it recall? And so here they kind of use this dread approach where they can turn on the astrocytes again after learning, and if the astrocytes are actually doing something long-lasting, if you come back after the learning and you activate the astrocytes, can you actually see a memory recall event? Which is cool. I mean, this is pretty crazy. So that's what they did. The animals were rejected with all these things. They do fear conditioning. They put them in a context B now, which should not normally induce recall. And then they add the drug that activates the receptor, the dread receptor in the astrocytes only. And then they see an increase in freezing, as if the animal was recalling that memory in the context that it had not done that before.
This is incredible. Because I remember when people first came up with this like FOSS-driven manipulation, it was all done in neurons first. So like you tag the neurons as previously active when you first learn the context is giving you a shock, and then subsequently you can make them out freeze even in a new context. And it was so new and so impressive, they called it Inception, because that's when the film came out.
Right.
And that was in neurons, but now you can get Inception also in astrocytes.
Right.
And they also found one of the side experiments actually showed that when you do this stimulation of astrocytes using these dread receptors, you actually can induce FOSS in neurons, in the engram neurons. So it gives you some potential mechanism of how this works, is that the astrocyte is triggering the neurons, promoting the neurons to fire, the engram neurons to fire again. Yeah.
Right. Yep. I skipped over that experiment just for time, but yeah, exactly. So there's crosstalk. So the astrocytes, I mean, we're sort of at the point now where we're saying, okay, there's a special set of astrocytes that are activated during learning. They seem to physically contact neurons that are active during learning, and they seem to specifically affect the synapses on those cells. And they're sufficient. So if you ignore the neurons, if you just activate the astrocytes only, that is good enough to change the properties of the neurons, including even inducing FOSS, which I would not have expected again to really see. So all of that's kind of surprising, and I think I would have been, as a reviewer, I would have been fine with all those experiments, but then they do a final sort of, I guess, cherry on the top. So that now they wanted to get more of the molecular mechanisms of how this is working. And they start with just looking at gene expression. So they wanted to see, okay, just what are the genes that are actually turned on in these astrocytes that are tagged with FOSS. And indeed, they show that there's a bunch of genes that are induced, and there's a bunch of genes that are downregulated. And so they just picked one of these genes to follow up on, and the one that they concentrate on is something called a nuclear factor IA, NFIA, which is a DNA binding motif, and I guess had been as a gene that in astrocytes had been implicated in regulating circuit function. So it made sense that they sort of wanted to concentrate on that particular gene. And so they, again, used their fancy genetics to specifically knock out that gene in astrocytes under the FOS promoter. So it's only the astrocytes that are activated where that protein is then, or that gene is deleted. And then they looked at, they looked at memory. So knocking out this particular gene just in the astrocytes, again, led to this decrease in freezing after recall, and a similar effect as you saw with the CFOS knockout. Although interestingly, that freezing effect in the recall was only seen, that deficit was only seen in the freezing with the contextual fear conditioning, but not in this novel place recognition assay. So now there seems to be some specificity of the kinds of memory that the astrocytes are actually regulating. And they sort of have at least one handle on one of these molecular mechanisms. Now, of course, the big question here is what is this NFIA actually doing in the astrocytes, and how does it regulate whatever the astrocyte is signaling to the neurons? And of course, that's a lot of follow-up experiments to sort of get at that exactly. So kind of a cool paper, very surprising results to the field in that this it's not so surprising that astrocytes are involved in memory processing per se, but the way that the astrocytes seem to be regulating memory looks very neuronal-like. It's using the same gene expression induction. They seem to then talk to the neuron talks to the astrocyte. So there's some sort of feedback loop here that the astrocytes are actually important for in terms of setting up the circuit. So now we have to sort of include astrocytes in the engram circuit because if you just activate the astrocytes, you can get recall of the memory. Now, I think I left out a control here, which was important to say that you still need neurons for the memory recall. So you can't just activate the astrocytes without activating the neurons. The neurons are if you silence the neurons and activate the astrocytes, you don't get recall.
Yeah,
so it seems to be... What's that?
They actually did the opposite experiment where they silenced the astrocyte ensemble, but then still activated the neurons. This is like the very last experiment they did. Activated the neuron engram, and they can get freezing back.
Right.
So it suggests that the neurons are kind of downstream, so to speak, of the astrocyte.
Right.
Yeah, which makes sense.
Exactly. So the neurons are still sufficient in themselves, and so the interpretation there is that the astrocytes are talking to the neurons, like that the neurons are still storing the information, but somehow the astrocytes are important for helping them get reactivated in some way. This is where there's a lot of speculation that you could sort of go on about what the astrocytes are actually doing to induce the recall event. But the fact that you can just activate these astrocytes only and then see a recall event is pretty wild.
So what do you think, Jason? What are the astrocytes doing? Do you have some ideas?
Yeah, I think so this comes down to like how is the engram formed, and so you have these synapses that have to be maintained physically to have the recall event over time. So my feeling is that the astrocytes are somehow involved in making that maintaining that physical change, whatever has happened, whether the synapse got bigger or if it made a new synapse, that the astrocytes are integral to that physical change. They're maintaining it, and that because they're also talking to the synapse in some way, and that part I don't understand. So it makes sense that the astrocytes are there to help stabilize that synapse, but why you would actually have the astrocyte talking to the neuron during that recall event, it's not clear to me.
So how are the astrocytes and neurons physically associated? Do we know?
So the astrocytes, so you have the pre and postsynaptic terminal, and then the astrocyte actually wraps around the whole thing.
Oh, okay.
And so when you look at EM reconstruction of synapses, most of these synapses have the three components, where there's the astrocyte enveloping the whole thing, and there's glutamate transporters, for example, on excitatory synapses in the astrocyte. So it's thought that, as I mentioned at the beginning, you get glutamate release, and then these glutamate transporters on the astrocytes help buffer and take the glutamate out of the synapse cleft. So there's certainly different ways. But what we don't know here is, so there's physical contact, but how does the astrocyte talk to the synapse in a way that it's instructive? So like there's some sort of calcium signal in the astrocyte that induces some signal now to the neuron and to the synapse, and that's not clear what that is either.
Do you think if we were to somehow kind of mess up the astrocyte signaling, so all of the astrocytes are now hyperexcited, do we form worse memory? Do we get better memory? Are things more mixed together because we are now less selective in our memory formation? Like how?
Yeah, I think most likely yes. That if all the astrocytes are sort of, if there's calcium misregulation in those astrocytes, that the stabilization of the engram. My feeling is that this is more of a, after learning thing that they're involved in versus just making, forming the circuit. But yeah, and there's known to be, in a disease context, these astrocytes get activated just like microglia, where they become physically different. Their physical contacts are different. The shape of the astrocyte is different. So one could imagine that some of the dysfunction that you see in these diseases is not just because the neurons are being disrupted, but the astrocytes are.
And what do you think, are they involved in the other side of plasticity, which is long-term depression?
Yeah, there's certainly evidence for microglia to be involved in depression, LTD,
in terms of
where it's thought that they can actually physically remove the receptor. And so there could be this sort of, but there is now evidence that maybe other glia do something similar when they take up, bits and pieces of synapses. But yeah, I guess we'll have to see. This does open up a lot of questions about the role of astrocytes in plasticity again.
Amazing work. Well, as you say, fancy genetics. Oh my gosh.
The genetics is absolutely.
Oh my gosh.
I feel like next flow chart to see what is talking to what.
Yeah, you look at their plasmid and virus methods, and it's just like a whole massive list.
It's amazing what you can do. And I want to remind everyone, they're using adeno-associated virus vectors, and the construction of those is infectious disease research. So if you want to have some neuroscience research, you better let that go.
That's absolute workhorse for neuroscience, AAV.
Yeah, it's cool. Who knew? That's TWiN 56. You can find show notes at microbe.tv/TWiN. If you have any questions or comments, twin@microbe.tv. If you like these programs, we'd love your support, microbe.tv/contribute. and write your senators to oppose nomination of RFK Jr. as head of HHS. www.safecommunitiescoalition.org/RFK. Jason Shepard is at the University of Utah in Salt Lake City. Thank you, Jason.
Yep, thanks.
Tim Chung is at New York University. Thanks, Tim.
Thanks very much. Thanks, Jason. I'm going to look up ways of boosting my astrocytes now to help me recall memory better.
The other idea of what astrocytes are doing is metabolic. So they're taking in all the waste products from neurons and then processing them. So there are probably ways to boost that.
I'm Vincent Racaniello. You can find me at microbe.tv. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.